My career in medicine and pathology started over 20 years ago in this very lab, and it has been very interesting to see how patient care has evolved since then. We now speak of precision medicine, biomarkers, and targeted therapy as key ingredients required for optimal outcomes. Even though our efforts directly impact most medical decision-making, we remain a vague concept to many of our colleagues, administration, patients, and the public. With no conversation happening, we will miss opportunities to receive support in response to increasing demands. To begin this conversation, we can shed light on the workings of a modern pathology laboratory. At specimen reception, the collection of information begins. Patient information and clinical history, if provided, must be re-entered into the Pathology Laboratory Information System, or LIS, and linked to existing patient clinical encounters. In our hospital, requisitions remain paper-based, and some information is handwritten to be retyped by a transcriptionist. For the surgical pathology service, a typical day's work would consist of 75 cases, with a case represented by 1 to 8 specimens on average. Small tissue biopsies are received in formalin jars and transferred into cassettes. Care is taken to contain small tissue fragments. Larger specimens must be prepared and placed in formalin prior to dissection and sampling. Many pathology departments now employ pathologists' assistants, or PAs, to carry out the description, dissection, and sampling of complex specimens, referred to as grossing. In some instances, over an hour of PA's time is required to complete a case. For example, a breast cancer specimen can require X-ray imaging, and the report will include detailed documentation about the tumor's macroscopic appearance and location. Tissue specimens must undergo adequate preservation in formalin, referred to as fixation. Sampling is done in a systematic manner with tissue placed in cassettes. The PA's findings are transcribed using voice recognition software, although dictation onto cassettes remains a more reliable backup option. In addition to tissue or surgical pathology samples, cell suspensions and small particles of tissue can be obtained from needle aspirates, body fluids, brushings or washings, referred to as cytology specimens. Cytology samples can be prepared to produce slides directly for assessment. To facilitate the evaluation of a large daily volume of cytology samples, cytotechnologists are employed to pre-screen all slides and provide a preliminary interpretation that is later confirmed by the pathologist. Before tissue can be viewed on slides, tissue processing, embedding, and sectioning is required. Processed tissue is embedded in paraffin wax, an inert material that can be sectioned a few microns thin. Cytology specimens can also be embedded into paraffin blocks called cell blocks, greatly expanding their utility for additional downstream testing normally intended for tissue samples. Once fixed and embedded in paraffin, tissue and cell blocks can be stored for decades at room temperature and provide adequate preservation of tissue morphology, protein integrity, as well as DNA and RNA to a certain extent. On a typical day, specimens can generate around 600 blocks, with virtually all smaller biopsy specimens available for interpretation within 24 hours of receiving the specimen. After mounting blocks on a microtome, tissue sections are cut, floated onto glass slides in a water bath, and reprocessed to remove paraffin. Tissue can then be stained using a conventional stain called H&E, which provides visualization of cellular details for diagnostic purposes. Once cover slipped, slides are ready to be read by pathologists on their microscopes. In addition to H&E stained slides, immunohistochemistry is a critical ancillary technique used by pathologists to characterize abnormalities, classify tumors, and perform biomarker profiling. Biomarkers are cellular targets that carry predictive and prognostic significance for a patient's given disease. For immunohistochemistry, sometimes called IHC, tissue sections are prepared using glass slides and incubated with commercially prepared antibodies designed to detect specific proteins within tissue sections. By obtaining an immunohistochemical profile in addition to tissue morphology, a more complex analysis of a patient's disease is possible. Tissue undergoes a pretreatment step called antigen retrieval, which promotes detection of antibody targets in formalin-fixed tissue. Antibody detection and visualization of the result, whether positive or negative, is a protocol that can be automated in labs to increase throughput. Here is a patient sample containing advanced lung cancer that expresses a protein called PD-L1. Because of this positive result, the patient is now eligible to obtain a highly effective form of immune-based therapy. Similar to IHC, fluorescence in situ hybridization, or FISH, also uses tissue sections and often serves as a test required for biomarker profiling. FISH allows specific DNA sequences within cancer cell nuclei to be detected. The pattern and quantity of signals obtained under fluorescent light will reveal the genetic abnormality present. Here is a patient sample of breast cancer showing an excessive number of orange signals that indicate the presence of a gene called HER2. While normal cells contain 2 copies of a gene, these cancer cells contain a much higher number, and this positive result indicates that the patient may benefit from targeted therapy that inactivates HER2. Without biomarker tests, there is no way of predicting whether some therapies will be effective. For many cases, a pathologist must integrate interpretation of tissue morphology, immunohistochemical profile, and biomarker results to provide a final diagnosis that is meaningful for the treating physician. The time taken for a pathologist to render a diagnosis is highly dependent on case complexity. Some cases consist of a single diagnostic slide, while other cases may require 100 or more slides for diagnosis. On occasion, a complex case may require several hours to complete. In addition to providing diagnostic services to patients within the hospital system, many larger centers also provide consultation services and biomarker testing for the surrounding community hospitals. The pathology clerical staff accession outside case material and process requests by registering patient information into the LIS. At this time, much of this work here is paper-based and requires transcription. Once slides have been reviewed and reports completed, all materials are filed within a storage area. Older cases are sent to a long-term storage facility. Molecular analysis of many cancer types has become standard of care worldwide. Within the last 2 years, our patients have benefited from the availability of next-generation sequencing, or NGS. NGS is a larger scale technique that provides DNA sequence information for the purpose of biomarker profiling of cancers, such as detection of mutations. Tissue arrives at the molecular lab on slides or in tubes. To increase sensitivity of detection, some samples are macro-dissected to enrich the areas of interest. Tissue is then dissolved and the DNA extracted. NGS requires that the DNA be fragmented and formatted, referred to as library preparation, in order for sequence analysis to occur. Sequence analysis is automated and currently requires around 1 working day to be completed. Once sequence data is obtained, comparison is made against a reference of normal DNA and any differences interpreted and reported. For this lung cancer patient, a deletion is detected within one area of the EGFR gene, called exon 19. In the era of targeted therapy, we know that there are effective treatments for lung cancer patients with this abnormality. Every day in our hospital and clinics, hundreds of patients undergo investigation by means of our services. From this brief look into the world of pathology, it is clear that every breakthrough brings more challenges to overcome. Expectations are higher than ever.